So my name's Kayleigh. I'm a PhD student at Manchester, and I'm just going to give you a brief overview of my PhD project, which is developing a new model to study Aspergillus infections in people with cystic fibrosis. So just a bit of background then, in case anybody doesn't know what is cystic fibrosis. So it's an inherited condition in which the lungs and the digestive system become clogged with this thick, sticky mucus. It's a result of a mutation in the cystic fibrosis transmembrane regulator, or the CFTR gene. And mutations in this gene cause a loss of function of the protein. And then this means that it can't carry out its job. So normally this protein is involved in the transport of ions and water across the cell. And because it can't transport the ions as effectively, it kind of results in this thick mucus. So in a non-cystic fibrosis lung, you do have mucus, but it's kind of not as thick and dense. In the cystic fibrosis lung, you can see here there's a buildup, and this can trap pathogens and make it harder to clear pathogens that are inhaled in the lung. So my project then is to create a new model for this, and just a bit of background of what is a disease model. So it's a biological system that we use in the lab that mirrors a disease or shows some of the same disease processes. And this allows us to basically recreate the disease in the lab and allows us to study it. So some common models that currently exist or cell culture models. These are probably the most commonly used, and there are normally two types. So we have primary cells, which are cells that have been isolated from patients or animals, and then we grow them in the lab. Or we have immortalized cell lines. So these are cells that have been altered to become immortal, essentially, so that we can just keep growing and growing them. And these are probably more common just because they're easier to work with and they're less expensive. We also have then animal models. So for cystic fibrosis in particular, a lot of the common animal models are obviously mice, pigs, and ferrets. And the animal models, normally we have to alter the CFTR gene in these models. So most of them are knockout, which essentially you delete the gene and it no longer functions, kind of giving these animals cystic fibrosis. So if we already have models, then why do we need to make a new one? Well, this is my project. So the problems with our current models, animals, there is high variability. So obviously for these experiments, we have to use a lot of animals, and there's a lot of genetic variability between each animal. So it makes it hard to compare between them. There's also ethical concerns. So for pigs in particular, when we give them cystic fibrosis, it kind of results in problems with their digestive tract, which have to be surgically corrected before they can be used in these experiments. So obviously it's not very nice for the animals. Also with the ethical concerns, then there's a limited number of experiments that we can carry out. It has to be really refined, and we can't just use as many animals as we want for the experiments. There has to be a lot of thought behind it to really limit the number of animals that we're using in these experiments. And also there's the cost associated with them. So getting the animals and then also caring for them while we're using them. For the current cell culture models then, primary cells, which are the ones that we obtain from people, can be quite difficult to maintain. So it can be hard to keep them growing in the lab, and also to collect them from the patients. It's not a very nice experience. They're also expensive. So again, if they're hard to maintain, we can't use them for many experiments. So the cost per experiment is quite high. And again, kind of the same thing with the animals is there's a high degree of genetic variability. So for example, if we're using cells from patients, per patient, there would be genetic differences. And again, this would make it hard to compare across them. So my project then is to develop a new model, and it's funded by the NC3Rs, which is the National Centre for Replacement, Refinement and Reduction of Animals in Research. And their work is essentially to try and reduce the number of animals that are used in research. So with that in mind, then my model was going to be a cell culture model. And basically what we've done is taken non-cystic fibrosis epithelial cells, which are just the cells in the lining of your lungs. We've then deleted the CFTR gene. So we have a collection of cells, some of them are non-CF, and then the ones in the grey you can see would have the CFTR gene deleted, essentially giving them cystic fibrosis. We then identified these cells that had the CFTR gene deleted, and collected them all together and grew them up. So we now have a group of cells that essentially all have cystic fibrosis. And from that, then we've created what's known as an isogenic pair. So we have the non-CF cells and then the CF cells, and they are essentially exactly the same apart from the loss of the CFTR gene. So that reduces all of the genetic variability, and allows us to have a really good comparison between the non-CF cells and the cystic fibrosis ones. So once we've had our model in, what can we do kind of in the context of aspergillosis infections? One of the first things we can do is look at immune response. So we have our CF cells and we have our non-CF cells, and we infect them with Aspergillus fumigatus spores. From this, then we can do experiments and we can compare the cytokine release. So cytokines are basically molecules that are released in response to infection, and they can kind of call other immune cells to the site of infection to deal with the Aspergillus spores. Ones in particular in cystic fibrosis that we would be keen to look at are known as IL-6 and IL-8. They're two important ones in inflammation. We can also then look at the ability of our cells to internalize spores. So one of the main mechanisms in getting rid of inhaled spores, or one of the mechanisms in getting rid of inhaled spores, is internalization by the epithelial cells. So essentially the cells are able to take in the spores, and then once they're inside the cell, they can kill them. So again, we've taken our two cells and we've added Aspergillus, and we've used a machine called flow cytometer, which basically allows us to image the epithelial cells, which you can see here, and see the Aspergillus inside of the cells. And from this, we've been able to compare the ability of the cystic fibrosis and non-cystic fibrosis cells to internalize the spores. And we found some really interesting stuff with this, in particular that the cystic fibrosis cells aren't able to do it as well, which could kind of indicate that that could be one of the reasons why they're not able to clear Aspergillus as effectively as people without cystic fibrosis. And lastly, then we're also looking at the ability of the two cell types to kill Aspergillus spores. So again, we've infected our cells, and then we can compare spore survival. So we infect with a known number of spores, we leave them with our cells to kill them for a certain amount of time, and then after we can grow out the spores and count them and kind of compare how many are able to survive. And again, this allows us to directly compare the ability of the cystic fibrosis and non-cystic fibrosis cells to kill the spores and get rid of the Aspergillus. So that's just kind of a small overview of what we've done so far. We have other things planned, obviously, but just thank you for listening, and just want to say thank you to my supervisor, Paul and Sarah, and the MFIG group. And yeah.
Thank you, Kayleigh. That was interesting. Do we have any questions?
We don't know.
Okay, I have one. So you've generated these two cell lines. Do you have detailed information on the genome, so you can compare why one is more able to kill spores than the other?
So we've made a knockout in A549s, and we've just knocked out the cystic fibrosis, the CFTR gene.
Okay, so it's something to do with the CF gene makes. Have you any idea why that might be happening?
Of why they're less able to internalize the
Yeah,
spores.
yeah.
No, that's something we're looking into. We don't know whether maybe it's something to do with when we've knocked out the CFTR gene, we've seen differences in the production of mucin. So we don't know whether maybe that could be contributing to their ability to not internalize, or if it's something to do with recognition. We're not sure.
Okay, so the cells are maybe able to produce less mucin. Are they sort of in suspension culture? Are you growing them in a layer so you can see the mucin forming?
Just a monolayer, and then we've collected the supernatant and stained. We have specific mucin antibodies here, so we've stained with those.